this will be the last meeting for our events, 3-in-1 programs. Let me introduce myself. My name is Azeri Gautama Arifin, and I will be the MC and moderator for today's lecture. Selamat siang, selamat datang di International Guest Lecture 3-in-1 program yang bertemakan Plant Biotechnology, a Modern Approach for Better Crops. Perkenalkan, nama saya Azeri Gautama Arifin, saya bertugas sebagai MC dan moderator untuk kuliah tamu hari ini. Kegiatan hari ini akan menjadi kegiatan terakhir untuk acara 3-in-1 pada tema ini. The Excellency, the Head of Department of Agronomy, Dr. Nurahmi Ardiarini, the Head of Plant Biotechnology 3-in-1 Program, Professor Arifin Sugiarto, Professor Jean Kallerhoff from University of Toulouse, Lecturer of Plant Biotechnology, and also the participants. Thank you very much for your coming on International Guest Lecture 3-in-1 Program. Last week, we learned about DNA recombinations, and today Professor Jean Kallerhoff will be sharing with us her expert opinion on DNA amplifications. Terima kasih kami sampaikan kepada Profesor Jean Kallerhoff dan Bapak Ibu dosen Fakultas Pertanian dan juga para peserta yang telah mengikuti International Guest Lecture 3-in-1 program. Pada minggu lalu kita telah mendengarkan penjelasan Profesor Jean mengenai rekombinasi DNA. Dan hari ini beliau akan menyampaikan materi mengenai amplifikasi DNA. Okay, without further ado, ladies and gentlemen, please welcome Professor Jean Kallerhoff.
Thank you, Azeri. Good morning to everybody.
Good morning.
So today, I will make a follow-up of last time's lecture and continue with the study of nucleic acids. And afterwards, we will see how proteins are studied. Now, you must take these lectures as introductory lectures, right? I have based myself on gene cloning in bacteria, and these techniques are largely applied, and you will use them all the time throughout your studies, and if you go later in labs to work. So it will be applied very often and on regular basis. So today we're going to see if this works. How nucleic acids are purified. So you can purify nucleic acids, either DNA or RNA, from any living tissue, be it plant, animals, bacteria, or yeast, right? The method will depend on the starting material. Normally for plants, it's a bit, it's more difficult because in plants you have lots of pigments, right? And these interfere a lot on the purification of nucleic acids. So the principle is quite simple. In tissues, you have DNA, proteins, sugars, lipids. You've got lots and lots and lots of different macromolecules. If you want to isolate nucleic acids, you need to get rid of all interfering material. So what we do is we grind the tissue and we will separate the proteins and cellular debris from the nucleic acids. And to do this, we use phenols. Phenol is a very, very, very strong organic solvent, and it will disrupt all proteins, right? And therefore, when you grind and you add phenol and you centrifuge, you will have two phases: an organic phase, which is down, right? An aqueous phase, which is in the supernatant, and in between both, you will have a thick layer of fluffy white thing. So the proteins will be in this, in between the aqueous phase and the organic phase. And the nucleic acids, DNA and RNA, will be in the aqueous phase. So this is what is of interest to us. It is the blue supernatant. So the supernatant is recovered, right? And we will first precipitate this aqueous phase. We will use salts, and the salts will precipitate the DNA and the RNA. So you will have a mixture of DNA and RNA. So now, if we want to have only RNA, we will treat the mixture with an enzyme called DNase, the deoxyribonucleic acid. DNase is an enzyme that will disrupt the DNA, and it will leave you the RNA in the supernatant. Now, if you want to obtain DNA, you will do the opposite. You will treat the mixture, DNA plus RNA, with an enzyme that will destroy the RNA, and you will be left with DNA. Right. When you treat the mixture of DNA and RNA, if you treat it specifically with a salt that is called lithium chloride, this will selectively precipitate the RNA. Now, these nucleic acids can be visualized after separation by gel electrophoresis. There are different ways to make gel electrophoresis. The most common for nucleic acid is to use agarose gels. Now, in this blue part, you have a gel where you have three major big bands and two faint ones. These have been treated by lithium chloride, and you can visualize here the RNAs. So you can only see ribosomal RNA and transfer RNA, right? Because these form the majority of RNA, right? You will not see messenger RNA because these are present in very, very minute quantities, about 0.1% of the total RNA. And therefore, to visualize the RNA, you will need specific probes. We learned about probes last time. That is you will use a specific probe containing a specific sequence, right, that has been labeled with either a radioactive label or a non-radioactive one, or by chemiluminescence. Okay, this will allow you to detect messenger RNAs. But for the moment, we are not going to see this. Now, here is a DNA blot, right, with different bands. You can see that you have a majority of bands that do not migrate. These are high molecular weight DNA. And these are smaller bands that have been cut, and therefore you can see smaller bands, right? So all you have to remember is that if you want to purify nucleic acids, you grind your tissue, you treat with phenol. The phenol will separate the nucleic acids from all the proteins and the debris, right? And you will recover the nucleic acids in the aqueous phase. And afterwards, you precipitate and you can work with these nucleic acids. If you want RNA, you destroy by DNA, you use DNAs. And if you want DNA, you treat it with RNAs. Okay, so once you've got your purified DNA or RNA, you need to measure it, measure the quantity. To measure the quantity, we are going to make an absorption curve. And this is the absorption curve of a typical nucleic acid, right? And you can see that there is a minimum at 230 nanometer wavelength, a maximum at 260, right? And the curve drops down with the absorption at 280, less than the one at 260. So to measure the concentration of DNA or RNA, we will use the absorption at 260 nanometers. And there is a correlation between the optical density at 260 with the concentration. So for a double-stranded DNA, an absorption ratio of 1 will give you 50 micrograms per milliliter of DNA. For a single-stranded, the relation is optical density of 1 equals 40 micrograms. And RNA is also single-stranded. Therefore, an optical density of 1 will correspond to 40 micrograms per milliliter. Normally, you need to know this, but normally when you have an exercise, the lecturers, the professors will give you this data. Right, so that's the first thing to remember. The second thing to remember is that we quantify the purity of DNA by measuring the absorption ratio between 260 nanometers and 280. You can see that for a nucleic acid, the optical density at 260 is high. And therefore, the optical density of a nucleic acid will be high. Normally, for a pure DNA or RNA, we say that if the ratio between the absorption between 260 and 280 is between 1.8 and 2, then you have pure nucleic acids. If the ratio is less than 1.7, this means that you have high protein contamination. Why do we say this? It's because proteins absorb much more at 280 than at 260. So this is something that you need to know. Okay, so if you want to quantify, to see if your DNA or RNA is pure, you measure this ratio 260 to 280, and if it is between 1.8 and 2, then you know you've got pure DNA and pure RNA, and that it is not contaminated by proteins. Okay? So now, how are we going to assess, to detect DNA sequences? Your Professor Afif asked me to speak about this technique, and to assess, to detect DNA sequences, we use something that is called, a technique that is called Southern blotting. It was discovered by a researcher named Mr. Southern, right? So he named this technique after his name, and this is something that is used very, very regularly and all the time. It is the basis of all DNA detection technology. So the essence is simple. You have a tissue, you grind it, you extract the DNA, right? So this is the genomic DNA. And of course, with this, you cannot do anything because it's too much entangled. Therefore, you will cut it with restriction enzymes. Restriction enzymes are proteins that will cut specifically double-stranded DNA at specific sequences. So these restriction enzymes will give you little fragments, and with these you can work with it. You make an agarose gel. You run the gel by applying a current. DNA is charged, right? And therefore it will go from the positive pole to the negative one, right? And you will have all these fragments along the gel. And you can see that there are no bands here. We cannot visualize bands. This is because the DNA is so, you've got many fragments of DNA, and they are high molecular weight, so you cannot detect them. So to detect them, you will need to use a probe, a radioactive probe, like the techniques we saw last time. Okay. So Southern blotting is based, right? When you have the DNA, when you have cut it using restriction enzymes, you will migrate this on a electrophoresis agarose gel. So small fragments will be at the bottom of the gel and large ones will be here on the top. And this agarose gel, right, you will treat it with sodium with a base so as to separate the DNA strands. You will denature the DNA and both strands will separate. And afterwards, you will transfer the gel on a nylon membrane. And all the bands will come on this nylon membrane. It's exactly the same thing that we saw last time. Last time we transferred colonies on a nylon membrane. Today we are transferring bands, DNA bands, on a nylon membrane. Now, how do you know? How can we know if what we are looking for? If a band that we are targeting, if it is there. And therefore, to do this, we will need to work with a probe. That is, this is a probe. It is a DNA sequence that has been radioactively labeled, and it will recognize specifically its band, corresponding band on the gel. So remember, the probe has to be labeled so that you can see it. So you take this probe, you will denature it so that it is single-stranded, right? And therefore the single-stranded will recognize the single-stranded on the membrane, and they will hybridize. And because your probe has been labeled, you can see it. So this is the probe base pairing. That's the probe. It recognizes specifically any DNA that is complementary to it. And if you expose it to an X-ray film, right, you should get, if my thing works, oh la la, voila. After exposing to an X-ray film, you will get bands. So this individual B has got one copy of the probe, of the corresponding probe, right? And here you've got two copies. This means that you have two genes that are complementary to the probe. Now, if you know the nature of the probe, you can detect, you can identify what gene sequence is in those bands. So probes, you always need to know what is the nature of the probe, okay? So if you are looking for a specific gene X in your DNA, in your genomic DNA, you will take a probe that corresponds to the X gene, and therefore you will detect each time you have got a signal on the bands, on the film, you will know that you have this X gene is present. There is a question here. Okay. Right. Oh, Azeri, I forgot to transfer you the PowerPoint.
Oh, yes.
Okay, I will do it.
Maybe later. Yes.
Yes. So to summarize this, right? Here you have the DNA gel after electrophoresis. Okay, so after electrophoresis, you transfer it to a nylon membrane or nitrocellulose. And for this to be able to transfer, you will put it in an ionic medium, right? And then cover. Here you've got the gel. Here you've got the membrane, right? And you cover this with a stack of paper towel, and you put a load, something like 500 grams, and all the bands that were in the gel will be transferred to the nitrocellulose, as you can see here. And the agarose gel is now white, no bands present. All the bands have been transferred to the membrane. Once this is transferred to the membrane, you can incubate it with your probe, and the probe is, as I told you, it's like a drone. It will look for its complementary sequences, and they will hybridize, right? Because complementary sequences hybridize together and to form a double DNA band. And this, you will be able to visualize this by autoradiography, right? Each time there will be a band, this means that you would have your DNA sequence that is corresponding to the probe. Okay? Azeri, do you think it's clear to the students?
Yes, I'm clear. Yes, it's clear. But maybe I will ask them.
Yes.
Apakah ada pertanyaan atau mungkin dirasa kurang paham tentang materi Southern blotting ini?
Oh, sorry, ma'am. I think saya masih belum paham, ma'am, yang ini.
Yang di bagian mana mungkin bisa?
Ya, mulai dari ini, ma'am. Saya rasa kemarin pas praktikum nggak ada bagian ini juga sih, ma'am.
Iya, karena ini nggak ada di praktikum ya. Okay, she said that she is not quite understand about this particular slide. So maybe please.
Thank you for asking, because
yes,
it is very important, because it is the basis of all what we call the DNA chip technology, right? And there is something that you need to remember, all of you. Molecular biology is a very easy thing. It is based on the properties of DNA. DNA, genomic DNA, is double-stranded. And because it's double-stranded, there is complementary base pairing, right? You can allow for mismatches, but it will be fixed like this, okay? One strand will be complementary to the other, right? And this is the basis of the Southern blotting technology that is used to detect all DNA sequences, the majority, right? So when you've got DNA, this is, remember the metaphasic chromosome? This is a bundle. It's like it's like a woolen ball, okay? You cannot do anything with big woolen balls or cotton balls. To work with it, you have to loosen them, okay? And to loosen these DNA chromosomes, you will use some special proteins that will cut this huge bundle of DNA into small fragments. So these small fragments represent the whole genome, right? But in small pieces. When you have small pieces, it is easier to work with them. So these small pieces, you are going to separate them by applying an electric current. The small ones will migrate fast, and the big ones will stay at the top. Okay? So once you would have done this, you will transfer all these bands to a membrane. Membranes can be of different types. They can be nylon, they can be cellulose, nitrocellulose. They can be different. Okay, so once you have transferred the bands, okay, you will label them, right, using a specific probe. The probe will go and look for the complementary sequence on the membrane. And because the probe is radioactively labeled, it will look for its complementary sequence and hybridize to it and reform a double-stranded DNA that you can visualize using an X-ray film. So has this been clear? There was a lady who asked the question.
Me, ma'am. Okay, ma'am. Thank you for your explanation. I think it's more clear.
Okay, so now this is summarized in this slide. So DNA extraction, you digest, you cut into small pieces using restriction enzymes, okay? And all the DNA is on the agarose gel. You transfer this on a membrane. This is the way you transfer, that is, you use a solution of high ionic salt medium, right? This will promote the transfer of the bands from the gel to the membrane. And you can see here, after one night of transfer, the gel that was dark here, it's light. All the bands have been transferred to the membrane. Once you've got this, it becomes easier to work with it. So this membrane, you will hybridize it to a probe, right? Both have been denatured, and the denatured probe, the single-stranded probe, will look for its complementary sequence on the membrane, and you will see the spots, the bands, using an X-ray film. Okay, is this clearer now, Inda?
Yes, ma'am.
Okay, so that's a very important slide for you to remember. And if you want to detect RNA, it will be the same thing, the same principle, except that you will isolate high molecular weight RNA, right? You will transfer it. You don't have restriction enzymes for RNA, right? This is the only one thing that differs. you transfer the RNA from the gel to the membrane, right? And then you use the membrane to hybridize with a specific probe. So it's essentially the same thing. It's the same principle, except that you load RNAs on your gel instead of loading DNAs. And RNA is single-stranded, right? So you don't need to denature it. However, the probe needs to be single-stranded. Therefore, you denature the probe. So the single-stranded DNA or RNA will go and recognize specifically its complementary sequence. Okay? Right. So the second important macromolecule in a living cell, these are proteins. Proteins are important tools as well when you want to work in molecular biology. So, as the same thing as we did for nucleic acids, we are going to see how we isolate and purify proteins. You must not mix the two techniques. They are different. Okay, so that's the first step. You grind in a convenient extraction buffer. When I say convenient, this means you need to adapt the buffer according to the contents of the tissue. For plants, you need to use solvents that will remove all the pigments like chlorophyll or lycopene and all this, right? And then you will separate the mix of proteins according to their properties. You can separate them according to the size, to the charge, or to the degree of hydrophobicity, right? So with the mixture of the proteins, right, you will precipitate the proteins because you have large volumes of buffer. To be able to work with them, you have to concentrate them. So if you have a big volume, say 10 milliliters or 50 milliliters, you have to reduce the volume to get it into a very small amount. And the readily used technique is to increase the ionic strength, right? Using ammonium sulfate precipitation. This will cause the proteins to precipitate. Or you can also use polyethylene glycol. So both techniques will force the proteins out of solution and they will precipitate. Once they are precipitated, you can work with them, you can purify them. You need to purify them because otherwise you are going to work with mixtures of proteins and this is not good. So the most studied method of proteins is again electrophoresis, just as DNA or RNA. You are going to separate the proteins according to their molecular weight. So how is this done? You know that proteins are charged. They can be positively charged, or they can be neutral, or they can be negatively charged. So what we do, we will we will convert these proteins into negatively charged proteins by treating them with a denaturing agent. This is called SDS, sodium dodecyl sulfate. It is negatively charged due to the sulfate groups, and therefore this molecule of DNA, which is big, will have large numbers of sulfate groups around the DNA. And one that is shorter, it will have smaller numbers of sulfate groups. And therefore, if you apply a current, right, small molecules will migrate fast and big molecules will migrate slowly. So therefore, you can see the bands, right? High molecular weight proteins up and small molecular weight proteins further down the gel. So this is one method of separating the proteins. So when you run a mixture of proteins on a gel, there will be about hundreds of bands on the gel, and you can't work with it. So we are now going to use something that you should know. It's Western blotting. So Mr. Southern discovered the DNA detection methods, and therefore he named it Southern blotting. To detect RNA, to differentiate between Southern and RNA detection methods, they named it Northern blotting. So for Northern, it's RNA. For Southern blotting, it's DNA. And to detect protein, the name of choice was to use the third cardinal point, Western blotting. So it's easy. You can remember this very easily. But here for the Western blotting technique, it's slightly, the strategy is the same. After running on the gel, right, you will transfer all these bands on a membrane, a nitrocellulose membrane. So all these bands will be on the membrane. So how do you know which one is which? There again, you will use a probe, but the probe will be different this time. Remember that these are proteins, right? So the probe that you will use will be an antibody. Have you heard of antibodies before? Mm-hmm.
Yes, ma'am.
Ah, good. Antibodies, this is what you get when you get vaccinated. Okay? So researchers take the protein, they inject it into an animal, mostly rabbits or poultry, and the animal will make a protein that will recognize specifically the protein, which is called antigen. So therefore, in this huge number of proteins, if you have a specific antibody, this antibody will recognize your protein, and there again, you can detect the antigen by using the antibody because it would have been labeled. It will become clearer in the next slide. So this is your antigen that you inoculate to an animal. So the antigen, you inject it into a rabbit. The rabbit will make a protein called an antibody that will recognize specifically and only this specific antigen. And here you will get a complex of antigen and antibody, right? So once it is complex, you will use an enzyme. An enzyme, you will fix it on the specific antibody, and the fact of sticking this enzyme to an antibody, we will get what we call a conjugate. A conjugated antibody is an antibody that contains an enzyme. Now, because you have this enzyme here, if you add a specific substrate to the enzyme will hydrolyze the substrate, and the product will give you a change in color. Therefore, each time you will have a change in color, you will know that you would have had you would have had an enzyme that is fixed to an antibody, and the antibody is fixed to the antigen. Okay? So this is a very powerful technique, right? That can be used also to screen recombinant bacteria that are specifically expressing a protein, right? So as much as we did this technique last week using detection of specific nucleic acids, we do the same thing, except that we will use an antibody specific for a protein. And when each time you have a protein in one of those recombinant bacteria, you will have a signal when you will, after hybridization, antibody antigen, and you will have a luminous signal when you put it on an X-ray film. So the technique is essentially the same for proteins or for nucleic acids. For nucleic acids, you use a specific nucleic acid probe. If you want to detect a protein, the probe will be an antibody. Okay? Is this clear to all students?
There is one question. Prof.
Yeah.
There is one question in the chat box.
I want to ask about the nitrocellulose paper to see the result. Does it need a computerized reading? Very nice question. If you have large amounts of colonies, large numbers of colonies, of course, you will have to put your nitrocellulose membrane on a photographic plate that will computerize the thing. But normally, because as I remember, to see the electrophoresis result, we need a gel dock for reading and getting the result. Yes, of course, if you have thousands of bands, you will need to have a special apparatus to read the different bands. But if you only have 10, you can do it just manually. Okay? Is this
So it doesn't need any computerized reading, and it just shown directly on the papers?
if you don't have large numbers, of course, you don't need computerized readings. But if you have all your blots, your results tightly fixed to one another, then you will have to ask a computer to make the difference. But normally, when you use a specific antibody, you should not get large numbers, because otherwise this means that you are getting unspecific reactions.
Okay, it's clear for me, ma'am. Thank you.
This means that your antibody is recognizing too many proteins. It is not specifically targeting your proteins. Okay?
Okay, thank you, ma'am.
Nice question. Thank you, Katya. So, as you can see, this is the antibody that will recognize specifically the antigen that is present in these recombinant clones, right? And therefore, you will know that in this clone, you have a bacteria that is expressing specifically the protein you want. So how do we that's again the antigen antibody. Okay, so how do we express proteins? You have two types of expression vectors that are based on the expression, on gene expression. You know that DNA is first transcribed into RNA, and RNA is translated into proteins. So you will have two types of expression vectors, and even three. You can specifically have transcription expression vectors, whereby you will have a promoter, and other regulatory sequence. Here you will have a multiple cloning site and a terminator. So because you have promoter and terminator sequences that regulate transcription, any sequence that is found in the multiple cloning site will be transcribed, and therefore you will have RNA, okay, because DNA is transcribed into RNA. Now, this is the well-known type of vector called transcription vector, PGM vector. Now, as all plasmids, it has got resistance to ampicillin. It has got an origin of replication. It has got a multiple cloning site, here with the multiple cloning site. Within, don't forget, the delta, the alpha peptide sequence of the LAC-Z gene. So you can see that all the lectures are linked one to each other. If you haven't understood the first and the second lecture, it will be very difficult for you to follow the third and fourth lectures, because I'm not going to go back each time. So this is the alpha peptide that corresponds to the beta galactosidase region. It has got the multiple cloning site, And here is the chromosomal DNA that contains the complement, the rest, the 3' end of the LAC-Z gene. So if you have no gene sequence within the multiple cloning site, this LAC-Z sequence will complement this one, and with the substrate of beta-galactosidase, right, your colony will be blue. If you have inserted a sequence, right, the colony will be white. Now, transcription vectors have, in addition, 2 promoters. The best known is a promoter from the bacteriophage T7, or what we called also the sp6 promoter. So the double-stranded DNA will be transcribed either with the T7 or with the sp6 promoter. And therefore, this will give you either a positive strand RNA or a negative strand RNA, depending on which enzyme you use. If you use the T7 polymerase, this polymerase will work with the T7 and will give you a positive or negative strand. And if you use the sp6, it is the reverse strand that will be read, because transcription is monocistronic, is done only on one strand. Okay, so with the transcription vector, you will get RNA. Is this clear? Okay, now, if you want to produce a protein, you will need an expression vector that will produce proteins. And the sequence of interest, right, is encompassed between a promoter and a terminator, but in addition, it has to have regulatory sequences so that translation is made possible. So the plasmid is always the same thing, selectable marker gene for selection of recombinant colonies and origin of replication, but for a translational protein expression vector, you will need, in addition, sequences so that ribosomes will recognize, right, initiation of translation. And here you will get both RNA and protein. And this is a very, very useful vector, because you will get within the recombinant bacteria, you will get directly the protein. Okay? So, this is another nice tool that is used in molecular biology, is the expression of proteins in bacteria. And these proteins are called fusion proteins. It allows the purification of proteins in high amounts, right? And you can use it for the obtention of high quantities of proteins in bacteria, right? And this is how many pharmaceutical agents are purified, okay? And this is based on the present the presence of a sequence that is linked to what we call a tag. A tag is a sort of signal molecule. And these two are separated by a protease coding sequence. Now, you must say, what is she speaking about? I don't understand. But you will understand now. If this bar can be here. So, you're going to express a sequence that is linked to a tag. What is this tag? There are many tags, but I have chosen the easiest one. So you have here the coding sequence that you want to express. Follow the mouse. This is first the coding sequence. Here you've got a sequence of 6 histidine, right? And between those two, you have a protease site. A protease is an enzyme that will cut proteins, that will hydrolyze at specific places proteins, just as much as we have endonucleases, exonucleases, right? We have protease that act on proteins. So remember that this is just the expressed, the targeted sequence. This is, of course, within a plasmid vector. When it is going to be expressed, all this will be formed in the protein. So you will have a protein that will contain the polyhistidine, the protease, and the protein corresponding to the cDNA sequence. So once you've got this protein, you will purify it. And this is the trick. You will run an affinity chromatography. On this column, you will have fixed a ligand. A ligand is a molecule that will specifically recognize your tag sequence. And here, for histidine, it is a chelated metal. So you have this column, and when you put this protein, the histidine tag will specifically bind to the polyhistidine, to the metal, right? And then it becomes a nice, easy game. The protease, right? Because this is a protein, that don't forget. The protease will cut specifically the protein, and you will get the purified protein in large amounts. Okay? I can see that it is not clear because I can remember that my students never understood this at the first instance. So, a fusion protein vector is a vector like this, okay? Because it will contain ribosome binding sites. And in the sequence of interest, on top of the sequence of interest, you will have, that's your sequence of interest, you will have a sort of a tag, a signal, and in between those two, you will have a protease site. So when you're going to get the translation, you will get here the polyhistidine, you will get the protease, and you will get the protein corresponding to the cDNA sequence. Okay, because all this would have been translated, right? So once your protein is produced, your objective is to get this red protein purified. So how are you going to do it? You take the bulk of the 3 proteins, you run it on an affinity chromatography, right? So the histidine will fix to the ligand, which is a chelated metal, right? So this is fixed through the histidine, and the protease is then activated so that the protease cuts specifically at the site, and it will give you a purified protein. Is this clear to everybody? Anybody has questions? Oh,
Yes, ma'am.
uh-huh.
Are the yes, the protease site is join the purified protein, because in this picture, I think the protease site is still join the purified protein, ma'am.
Azeri, I need your help.
Oke, Bu Azeri,
Ya.
jadi ini kan yang di purified protein itu terlihat bahwa ada yang warna birunya itu, ma'am. Itu kan terikut. Apakah memang benar terikut atau sebenarnya dari dari ini kan penghubungnya kan, ma'am. Itu apakah memang terikut atau tidak terikut, ma'am? Soalnya kan kalau purified protein setahu saya kan itu memang hanya protein saja tanpa ada pengikut lainnya. Terima kasih.
Oke, terima kasih atas pertanyaannya. Oke, Professor Jean, she asked about the below picture. I think there is 3 pictures, right? And in the right side, it's shown that the purified protein has a little bit blue.
Yes. Yes.
Yeah. And then she
Yes.
asked if it's purified, it should be all red, but why there is a blue in it?
Good question, my dear. Yes. Sometimes I could have made all this red, but life is not so simple as this, okay? Because sometimes the protease does not cut exactly at the linkage. So you can have a protein with 1 or 2 amino acids, but you can also have a protein that has no blue at all. It depends on the way you have designed your sequence. Here, I made a little bit of blue so that I show you that in biology, nothing is clear cut, okay? But this is not going to hamper, this is not going to affect the stability and the functionality of the protein, okay? So you can also have proteins without the blue. It depends on where the cleavage site is directed. Okay? Did I answer your question, lady?
Yes, ma'am.
Another question. Attendance form. Always the attendance form. They track you. You cannot just not attend the courses. Haha. Okay. I think I have gone. Okay, yes. So when you go and buy bread, sometimes you have incorporated, you use a specific protein, or if you want, sometimes in pharmaceutical industries. You need large amounts of proteins, right? So therefore, you need to produce these proteins industrially. So you optimize your system in the lab using small quantities, right? Once you have checked that your recombinant bacteria is producing the right protein that will do the correct function, right? Then you can scale it up. That is, instead of producing small amounts in 10 milliliter microplates, you can scale it up to 1 liter flasks, and you check that the protein is produced, or you can produce the same protein, right, using multiple small fermentation batteries, right? So don't forget that you started from one recombinant bacteria that is producing your specific protein, the one you want, okay? And so this recombinant bacteria, you put it into 10 milliliter culture, you check that your protein is being produced, then you scale it up, right? You can scale it up from 1 liter to 2 liters to 300 liters. But now what is also very commonly done is that you produce the same protein in micro fermenters. Okay, so this means that instead of using 300 liter fermenter, that is not sustainably accepted because it will use lots of electricity, lots of water, right? And lots of energy. And you know that energy is getting rare and rare and rare and very expensive. I understand that in Indonesia, it is also the same as in Europe because of this silly man who started this silly war, right? So we must all be very careful about the energy we use. What I'm doing here is that I'm using a lamp during the day. This is not good, but it's better for me because I can see better. So you understand that you can, after in the lab, you start from 1 milliliter, 10 milliliters, you can scale up to 300 liter. And of course, you produce massive amounts of protein. But if you want to be sustainable, you, instead of working in 300 liter fermenters, you work in micro batteries, right? So that you will use less water, less energy, and therefore but you will still produce the same amount, because this is what you're targeting, produce same amount of proteins as in your huge fermenters, right? But using less energy. Okay? Right. So I have reached, I did not prepare more slides for you because you would have been bored. And for the next 10 minutes, we will allow some time for questions. not only from this lecture, but from the first 3 lectures as well.
Okay, is there anyone who want to ask questions? Mungkin ada yang mau bertanya, bisa dari materi hari ini atau 3 pertemuan sebelumnya. Kami persilakan.
Are you sure you've understood everything? Haha. Your professors will tell me the results of your examination. So, for instance, how do you learn your lectures? You will scroll through the slides and say here. When you have one slide, you need to ask yourself, what is the message that comes from this slide? Okay, you have several messages to take home. First one, what solvent is used to purify nucleic acid? So the answer is phenol. So what is the principle underlying phenol extraction, right? This is based on the separation of an aqueous phase with a solvent phase. And the nucleic acids will be in the supernatant, and in the organic phase will contain all sorts of debris. So, and then what methods can be used specifically to isolate RNA or DNA? So you see, the only way for you to work on your lectures is to devise take-home messages. What do the lecturers want me to remember? And it is the same for courses with your professors at the UB. They also have slides with messages. Okay, so you do the same thing. Here, very important slide. How do I quantify nucleic acid, right? What is the principle of southern blotting? What is the principle of northern blotting? What are the important steps? Here in southern blotting, the important step is to have a denatured DNA strand fixed on a membrane and to use one probe that also has been denatured, and the probe will look for its complementary sequence on the gel. Okay, so you do this for all the slides, or for the main slides that your professors would have shown you. If I had to interrogate you, I would ask you, what is the principle of producing protein using a fusion protein vector? Aha. That would get you into trouble, okay? If you have not understood okay, Non, tout est figé là. Can you hear me?
Yes, we can.
Because on my screen, this says that Zoom has been interrupted. Okay. Right, no more questions? You've got 3 minutes.
Is there any questions? Anyone? Ada yang ingin bertanya?
So you should all
Oh.
have. There's one.
There is one question from Shakira Maulidia. I'm curious about this. Is chromatin topology important for gene expression?
Of course, because the chromatin will bind to the DNA only in eukaryotes, right? And this is what gives the DNA a tertiary and a quaternary structure. So, in fact, if you want to express a gene that is right in the middle of this chromosome, you have to disrupt it. And the chromatin, depending on whether the proteins are methylated or phosphorylated, it will be very important for gene expression. Not all proteins will be acetylated or methylated. Methylation is associated to gene silencing, that is, knockdown of protein expression. Okay, that's a very, very important question. Any more questions? So, okay, then I will close down and I will thank you for your attention and also thank your professors for giving me the opportunity of delivering some lectures to their students.
Thank you, Professor Jin. And because the time is already up and there is no more questions, before closing, I will introduce you to the head of our Department of Agronomy. There is a Dr. Nurrahmi Ardiarini, and she will give the closing testimony about this program. And also,
hello, Professor Jin.
Yeah.
Good morning, Professor Noor.
Good morning, Professor Jin. How are you?
I'm fine, very well. Thank you. Thank you very much for giving me the opportunity to
you're welcome.
Lectures in your department to students of your department. They asked questions readily, and I hope that they would have understood most of it.
Yeah, I hear for the 1.5 hours. Yeah, 1.5 hours. Yeah.
6 hours in total.
Yeah.
Okay, ladies and gentlemen, now we are going to listen to the closing testimony delivered by the head of Department of Agronomy, Dr. Nurrahmi Ardiarini. And afterwards, we would like to request the head to deliver the certificate of appreciations to Professor Jin Kallerhoff. Ladies and gentlemen, please welcome Dr. Nurrahmi Ardiarini.
Okay, thank you, Azeri. Honorable Secretary of Department Agronomy, Dr. Afifuddin Latif Adirejo, Honorable Head of Plant Breeding Laboratory, Professor Arifin Nur Sugiarto, Honorable Professor Jin Kallerhoff, as speaker from University of Toulouse, France. Dear colleagues and students, Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, as a participant at the 3-in-1 case lecture and a title of Plant Biotechnology, a modern approach for better crops, I am pleased to speak to you at the closing of the event. I believe that a productive progress has been made. This guest lecture, an excellent example of successful cooperation. I am grateful to you all for being part of this effort. Thank you for all the speaker, moderator, participant, and the committee that progressing this important event to a successful conclusion. Thank you for taking part and contributing this success. We say Alhamdulillah, I now declare this event officially closed. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, we give applause.
Thank you very much.
Thank you, Professor Jean.
My pleasure.
Please for the IT Mas Dian to share the certificate.
Yeah, this is appreciation, right?
Thank you very much. Thank you very much, Professor.
Thank you very much, Professor Kallerhoff, right? Kallerhoff.
Yes, yes. Pleased to meet you. And
Nice to
sorry
meet
to
you
have
too.
heard the earthquake in near Jakarta.
Yeah.
Thank you very much. Maybe from Afif, if you want to.
Professor, when you come to Malang?
Oh. I hope to come. I don't know. I don't know. Hopefully.
Maybe you can ask Afif.
Yes, yes. I can come and deliver more lectures. Yes.
Yeah, I hope so.
Yeah, the times when you come here in 2018, I think, right?
Yes,
you visit,
19.
yeah. At the time, Dr. Rahmi, she was not here. Yeah, because you went to another city, I think, Rahmi.
Yeah.
Yeah, with all of the college of Department of Agronomy, you know. At the times, Prof Jean come to our campus.
Yeah.
But we were not here. Yeah, what city? In
Yes,
2018, right? 2018. Yeah.
2018?
Yeah,
yes,
yeah.
yes.
You deliver some course. No, not
Yes.
Biotechnology, but in general, in front of our students at the time in sixth floor. Yeah, sixth floor of the building of GS times. Yeah. You met several colleagues like Dr. Cahyo.
Yes.
Yeah, you remember him?
Yes, of course.
And Dr. Karuniawan.
Professor Cho?
No.
No.
Professor Ifan.
Oh.
Because at the time he was the head of our international office. And also, Prof. Jean, you were head of international relation of INP Toulouse, right? At the time.
Yes.
Yeah.
Yes.
Yeah,
But
sometimes we hope you can.
Hopefully, yes. Hopefully.
Yeah. Okay, Azeri.
Thank you very much, Azeri.
Hi, this is
everybody.
Very nice for us.
Thank you very much.
I give back to you, Azeri.
Okay, ladies and gentlemen, we have had productive and inspiring time together, and this lecture about to come to the very end. I hope you have found the lecture on this 3-in-1 program are informative. Thank you very much. May all of you have a great day. See you.
Sorry, Bu Azeri, I interrupted you. Maybe we can take a
picture. Oh, yeah.
Picture, yeah.
Yes. Can you
please
on cam, everybody,
and
we will take a picture.
Yeah.
Yes.
Full souvenir.
How are you going to take this picture, Azeri?
By screen.
We will print screen.
Okay.
Yeah, everybody should be on camera.
Ini harusnya semua full toh tampilannya.
Yeah.
Bisa dibuka kameranya buat student.
Mas Dian, ini harusnya bukan gini.
Ya,
tampilan.
ya. Tampilannya bukan anu Pak Dian. Tampilannya bukan speaker. All.
Remove spotlight.
Oke.
Uh,
Halo, student, biar
Mungkin
segera
Mas Dian
ditutup.
bisa delete.
Sudah.
Sudah.
Oke, mungkin semuanya bisa on camera, on cam untuk dokumentasi. Iya. Oke, untuk slide pertama 1, 2, 3. Oke, untuk yang slide kedua 1, 2, 3. Oke, Mas Dian. Oke, terima kasih. Thank you everybody. See you.
Thank you.
Thank you.
Thank you.
See you, Professor Dean.
Bye-bye.
Bye bye.
Bye.
Je vous envoie le mail.
Oui, je